the Ewing 99 that is nearly closed, the Ewing 2008 in German countries, and the Ewing 2012 that has opened in UK and should open quite shortly in France. Which is good, is that medical doctors have been very good students. They have understand that this disease is a TIA disease, and all these trials are open 5 to 40 or 50 years old. But there is no much improvement for the last years in bone sarcoma survival. And the main problem remains for newly patients with metastasis at diagnosis, for patients with localized disease and poor response to chemotherapy, and for recurrent bone sarcomas. Have we done all we can do with chemotherapy? I don't know. There is some studies saying that maybe those that have more side effects with methotrexate may be more sensitive to chemotherapy, then maybe we should continue to explore pharmacogenetic of this disease. But still, we have to find a way to improve the survival, and it can be quite tricky. And for that, understanding biology of the tumor is quite important. But where to start? Because a malignant tumor is not just a tumor, it's a tumor with a lot of characteristics. It grows, it resists to death, but it also escapes to the immune system, is able to induce angiogenesis, and is not on its own, it's in an environment, and especially in bone, is in a bone environment. Then we thought that maybe Ewing should be more easier because there is a causal abnormality to the disease that will induce all the phenotype. And that targeting this abnormality, this is a fusion protein, might revert the malignant phenotype and help to control the disease. And indeed, if you do it in vitro or in vivo with small molecules like oligonucleotide, RNA, antisense, or siRNA, you have a very good efficiency. But this is not usable in human. And try to target this protein is just a nightmare. There have been some hope with a chemotherapy agent. It is not really new, but that were supposed to target more specifically disease with translocation as in Ewing sarcoma. But the phase 1 and phase 2 trials are not very impressive in terms of efficacy. Then efficient, but not easy. Then we have to find other ways. This protein is a protein that will be, that is a transcription factor, and it will link to the DNA and help to express other proteins that will be responsible of the phenotype. To that, they work in a complex. They don't work, the transcript doesn't work alone. Then you can also target this complex, and there are also in vitro assays that are efficient. There is also a new drug that is efficient in vitro and in vivo, but as you can see, the preclinical work has been done in 2009, we are in 2014, and for the moment, there is no trial going on with that drug. Then we can go further down and try to inhibit the different pathway that will be responsible of the phenotype. Among this pathway, one has been a source of great hope during few years, which is the EGF-1R pathway. It's a growth factor, and it has been shown that this pathway was activated in Ewing sarcoma, and it was responsible of the phenotype. When you inhibit this pathway, then you can revert the phenotype and have a great efficacy in vitro, inducing apoptosis, but not just in vitro. There have been many, many trials with different molecules targeting this pathway, and that was also a very huge hope, because some patients were really responding very well to this drug with partial response, as you can see there, even in patients with very, very important metastatic disease. Unfortunately, very few of the patients did respond, and it was quite impossible to understand which patients do respond and which do not, and impossible to predict who will be responding. The other thing is that effect was transient. If you continue to treat the patient with the IGF-1R inhibitors, at some point the disease reappears. Why that? Because just one receptor is not just one receptor. It's one receptor interacting with others and responsible of a lot of activation of downstream pathway. Then any of these pathway, any of these co-receptor can link to resistance by upregulation. Then the logical thing was to use combination, and several combinations have been used with different tyrosine kinase inhibitors, inhibiting different receptors, and by inhibiting the IGF-1R pathway at level of the receptor and at downstream pathway, like with mTOR inhibitor association. But at the end, which one is the right combination, we still don't know. And the great hope turned to a real despair. Real despair, because after so many trials, so many patients involved in these trials, the industry, the pharma, have stopped the development of these drugs. Why? Because, as you've seen, very few people respond. It was difficult to predict who will respond, and it was not useful for the main adult cancer that represent the mass of patients to treat, which means breast, prostatitis, and colon, and all of them. Then for the moment, what's still from this is that, what remains from this, is that there are some inhibitors from downstream, like mTOR inhibitors, or others that are still in clinical trial, but we don't know where it will go. There is another pathway that can give hope. That is the PRKC pathway that is overexpressed in Ewing sarcoma. And when you inhibit it, you are able to have activity in vitro and in vivo. But the inhibitor that we know now of the PKC, which is enzastorin, is usually developed in breast, prostate, colon, but no trial in Ewing sarcoma for the moment. Another hope, PARP inhibitors. This time, it has been a surprise, but a nice surprise. It has been a study trying to look at different screening drug sensitivity. There have been over 130 drugs tried in about 2,050 different cell lines from different cell types. And on the top of it, you can see it. The one that has shown very significant sensitivity here is Ewing sarcoma cells sensitive to a PARP inhibitor. And this was confirmed with other PARP inhibitors that are there, with a very huge sensitivity of Ewing sarcomas cell lines compared to other cell types. The same here. Which is PARP and what role it has? It's involved in DNA repair, DNA methylation, chromatin structure, and transcription, which means it's helps to repair DNA, but it helps also to express other proteins. Its function to the repair is that when there is a single-strand break DNA damage, PARP will recognize it and will bring the Baer pathway, that is a repair pathway, to repair these abnormalities. When PARP is inhibited, then what you can see is that this breaks still, and then a second break will occur and there will be a double-strand break that will lead to death by apoptosis. Why is Ewing sarcoma so sensitive? Maybe because the transcript of Ewing sarcoma is also involved in the expression of the PARP inhibition, of PARP. This DNA damage can occur spontaneously in a tumor, but can also be induced by different chemotherapy, especially alkylating agents that are used in Ewing sarcoma. Then combination with different chemotherapy have been tried. There is synergism with temozolomide with a huge efficacy when you use both drugs as compared to each drug alone in vivo, with complete tumor regression, including for metastasis, lung metastasis. Synergism was also observed with irinotecan, and these two drugs are two drugs that are used in relapse Ewing sarcoma. There is another combination that has been tried, but they didn't show any increased effect with PARP inhibition, probably because the combination already of chemotherapy was too efficient in these models, which is not always the case in patients, which means that the choice of model is also very important to translate what's happened in vivo and what will be in patient. And at the moment, there are several trials that are starting. Just one above 13, two about 18, and there is one pediatric trial below 18 that should start soon. The other ways to target Ewing sarcoma have been various. Angiogenesis is one of them, either with metronomic chemotherapy, which means small doses of chemotherapy that will try to interfere with angiogenesis, but also with the immune system. Some antibodies like bevacizumab that are currently in trial, and different multi-tyrosine kinase inhibitor with a specific angiogenic activity. Bone microenvironment is the next step for the next trial in the Ewing 2012, and it's already studied in the Ewing 2008. I will explain you with osteosarcoma why, because the mechanisms are similar. And there is one other specificity. You have heard maybe about antibody against GD2 in neuroblastoma that have bring a lot of hope for these children. This GD2 antibody is also, antigen is also expressed in Ewing sarcoma. And either instead of developing antibody, T-cell immunity is going studied. But how to integrate all these things to the current treatment? In first line, we already talked about some zoledronic acid, but in recurrence, for the moment, there is no standard. Then the next step is to have a standard chemotherapy for the relapse Ewing sarcoma, and there is a trial that should be open soon that is called RECURE. For osteosarcoma, it's much more complex. You have seen with Ewing sarcoma single abnormality leading to all that, and we are not able to target what you should target to be very efficient. In osteosarcoma, there is no one abnormalities, but there is like hundreds and hundreds, and it's a very complicated disease to understand, and I think that we don't really understand it yet. Which means that to try to target it, we have rather choose to target different things, and the microenvironment was the first things that we tried to target. When I talk about microenvironment, I talk about anti-tumor activity, immune activity, bone microenvironment, and angiogenesis. Then the first two ones, have been quickly introduced in phase 3 trial, but you should think it's a good thing, but you will see that Maybe it's more complicated than that. First, tumor-associated macrophages have been shown to be important either in Ewing sarcoma and in osteosarcoma. There is two kinds of macrophages, to simplify, the M2 and the M1. The M2 will favor tumor and the M1 will help to control it. The M2 is expressed in Ewing sarcoma and the M1 in osteosarcoma. Then there are some molecules that try to switch as much as possible to the M1 in osteosarcoma to try to control it. One of these is the mifemertib, or MTP, that you have probably heard about. That is a stimulator of the immunity, macrophage immunity. There has been a trial that has been very discussed that was randomizing a newly diagnosed patient, either localized or metastatic, at diagnosis between two different kinds of chemotherapy, MAP and MAPI, and randomizing also for MTP. Then there were 4 arms, 2x2 factorial design, which is important even if you don't understand, the MTP, even if randomized at diagnostic, was not started immediately. It was started just for maintenance treatment. The first result of this trial was published in 2005. The results were there is an interaction between chemotherapy and MTP, which means that statistically, with a 2x2 factorial design, we cannot interpret the results. Which was quite confusing, because when you look at the numbers, you wanted to say, okay, MTP is doing something. It has been a lot of debate, and FDA has said that with these results, it was not possible to allow the MTP. 3 years later, with the same result, just a little bit more follow-up, there were another publication. This publication says there is no interaction between chemotherapy and MTP, and there is an advantage to give MTP in association with chemotherapy, whatever the chemotherapy is. With these results, the EMA gave approval in Europe, which makes things more complicated for the rest of it. In this trial, I told you it was localized and metastatic disease, and the results have been published separately. Then this is just for localized osteosarcoma, which means that the approval of the EMA is just for localized newly diagnostic patient. For the metastatic patient on this trial, the tendency was the same, with maybe an advantage for MTP, but the number of patients was not enough to say that there is a clear advantage. There have been also a study in relapse or recurrent disease, and in that study, the kinetic was identical. The kinetic of MTP was identical between the youngest patient and the older ones. The tolerance was good, as in the previous trial, and they showed that it was clearly more efficient when the disease was minimal, which means that when there is no disease visible. With that, as I told you, FDA did not approve the drug, EMA approved it, but in France, which is Europe, the drug is not allowed. Then we are in a very confusing situation where some countries are using it, others are not. And for the patient, we still don't really know which is the benefit, even if we want to believe that there is a benefit. But the pharma that is developing this drug doesn't want to do further trials. When you look at what is going on now, there is a surveillance study, which means that if you treat a patient, you can follow up a form to explain what has happened to the patient, but this is not a trial. And there is another trial that is led in Italy, where the MTP is used for patients that have a very bad prognostic according to their prognostic factor, because they have shown that patients with high expression of PGP have a worse prognostic than others. There is a study that is supposed to open. I have heard about it like 1 year ago, and when I asked 2 days ago if it has opened, they told me, we don't heard about it. Then still waiting for it. About immunity, there have been another trial, which is the RAMOS-1 trial that maybe some of you have used. That was also in localized newly osteosarcoma. And for good responders to chemotherapy, interferon was used in maintenance treatment. Nearly 1,000 patients were good responders. They were randomized at week 12. 20% of them refused to be randomized, which is usual in a trial. But the treatment was starting only at week 30. And by that time, 23% of the patients did not started the MTP when they were supposed to start it. And along the treatment, mainly due to toxicity, also 55% of them have to stop the treatment earlier on. At the end, there were no statistical differences between the arm that was supposed to have interferon and the arm that was not supposed to have it. But it's difficult to conclude if there is no efficacy or if it's only a problem of compliance because the treatment was considered as too toxic by the patient that they didn't want to receive it. The other way to try to get over osteosarcoma is bone microenvironment. For that, there is a vicious cycle that is going on with you have a bone tumor. Tumor secretes rank ligand that will activate osteoclast. Osteoclast will induce resorption of bone. This resorption of bone and osteoclast will liberate growth factor that will make tumor grow, and it will going on and on and on. If you inhibit this pathway at different level, you will be able to decrease resorption and to decrease bone tumor. It will be able to prevent osteolytic lesion, to reduce tumor size and incidence, to inhibit migration, to reduce metastasis, and to increase animal survival. And there is some synergy with chemotherapy, especially with ifosfamide. But there is no effect with OPG and rank FC. There is no effect directly on osteoblastoma. When you have only lung metastasis and no bone tumor, there is no effect. And we don't know what will be the effect on bone growth. There is a drug that is a biphosphonate called zoledronic acid that has all these effects, but has also a direct effect on osteosarcoma cells. And we know now that the effect on bone growth is transient with that drug. This drug is the basis of the OS 2006 protocol in osteosarcoma and will be used also in the next Ewing sarcoma protocols. That's the same thing that I told you. The last characteristic of the tumor that has been targeted is angiogenesis. By different ways, but the one that is most ahead is multi-tyrosine kinase inhibitor, and especially sorafenib, that has been used by the Italians, with a huge effect in vivo, with anti-angiogenic effect and also with anti-metastatic effect. the clinical trial, a phase 2 trial in patients with recurrent disease, showed that partial response can be achieved, but in a very low number of patients, which means that having a huge effect in vivo doesn't translate in humans, and probably because the models that we have should be improved. The tolerance was quite good, but there are some effects that were not expected, especially skin effects, and that we have to learn to know. And there are also mycositis. They tried to understand why some patients were not responding, and they showed that the mTOR pathway was, the mTOR2 pathway was activated, and if you use an mTOR inhibitor, you are able to revert this resistance. But again, it's in vivo. What's about in patient? The trial is going on, then maybe we will have the results in few times. This trial is open just for adults for the moment, and they are thinking to do it in children. But the tolerance, especially mycositis, should be a problem because mTOR inhibitor also gives mycositis. Angiogenesis is the fun of the moment because there are several trials that will open or already open in osteosarcoma with different multityrosine kinase inhibitor that have all different activities, but they are all anti-angiogenics. There are many other targets that can be used in osteosarcoma, so many that I will not tell you everything, and all steps of metastasis can also be targeted. But all that, for the moment, are not really translated to someone useful for the patient. And in vivo and in vitro, you can say quite a lot of things. And for example, with HDAC inhibitors, you have trials, you have in vivo experiments saying that it's useful, and others saying that it's a problem because it's promoting metastasis. Then be careful. And as you see, there is very, very many targets, and then you can try to use broad spectrum new targeted agent. I'm not sure that we can call that targeting, but I like HSP90 inhibitors because they are targeting a target in each of the pathways that characterize the tumor. But if you look at clinical trial, then the results is disappointing. Okay, it's the first generation, and some have been optimized, but we don't know what it will give, what it will be for the future. There are other approaches like metronomic chemotherapy too. And the conclusion is that there is quite some more pieces to bring together to be able to revert the resistance of these tumors and to be successful in the drug development. We have to try to learn more about biology to find new ways to overcome this treatment resistance. You have to think efficacy, but at some points we will also have to think about late effects because we have no clue of what will be the late effect of these new drugs. And that's drug development in bone sarcomas. You have a young with a bone tumor. You have a very enthusiastic doctor that wants to move forward in the development and is so kind for that he's very proactive for banking tumor to be able to give them to a researcher also very motivated to try to understand the biology of this tumor and to try to find new targets. What we should do is bring pharma very early in the development of the drugs and in the process of discovering new targets to make them really involved in the disease. At the end, maybe we'll find new drugs, and then the medical doctor will have to be very enthusiastic to understand how to use them with the current treatment, and not forget to continue to bank tumor to see why patients are responding and why some are resistant. Then, at the end, what is the most important is to work together. And for that, ENCCA has been a huge help for networking in this area, especially between clinician and researcher, a little bit less with pharma. And there is a special work package for bone tumor that is the work package 7. The other thing is, we are here to talk about adolescents and young adults. Then be clever, continue to think of them. As you can see, the incidence is about 15 years for primary diagnostic, but the relapse will occur usually within the 2 years that will follow the diagnosis, which means that most of the patients at the relapse will have less than 18 years old. Then we should be a better students for protocol in relapse disease, because as you have seen along the presentation, most of them were only open for adults. But the bad students are not the medical doctors, because they have understand that. The bad students are the pharma. Then you have to help us, especially the young people that are directly concerned, and the parents have to help us to try to convince pharma and the authorities that this trial should not be started to adults, but should start much more earlier, at least at 13, especially because in adolescents there is no much difference for all these new drugs in the kinetic of these drugs. And thank you.